As you settle into stillness and allow your breath to slow, you might begin to notice how every part of you, from your skin to your thoughts, is quietly composed of countless tiny pieces, invisible yet essential. This calm journey begins in that invisible world, a realm where molecules drift, connect, and separate, carrying out the most delicate and vital tasks without ever asking to be noticed. In the study of biology, the science of life, we often begin by looking at cells, small, self-contained units that carry out all the functions required to live. But even these complex structures, remarkable in their own right, are themselves made of smaller parts. These parts are molecules, and the molecules of life form the silent foundation of everything we call living. They are not alive in themselves, and yet, without them, life would not be possible. Tonight, we begin not with creatures or organs, not even with cells or tissues, but with these smallest participants in the story of life, molecules. Simple, elegant, quietly doing their work. Each molecule is formed from atoms, the smallest units of matter. These atoms come together through bonds, attractions that arise from the delicate interplay of electrons to form stable groupings. When two or more atoms bind in this way, they form a molecule, and molecules, in turn, form the architecture of life. Your body, your surroundings, the food you eat, and the air you breathe. All of it is composed of these unassuming, ever-present arrangements. What makes the molecules of life unique is that they follow a pattern across every species, whether a towering tree or a small insect, whether the cells of your own body or those of distant coral. The molecules used in life are remarkably similar. This shared chemistry speaks softly of a deep unity, a quiet thread running through all living things. There are 4 major categories of biological molecules, sometimes called macromolecules, meaning large molecules. These are carbohydrates, lipids, proteins, and nucleic acids. Each plays its own part in the gentle choreography of life. And over the course of this lecture, we'll spend time with each one. But before we explore them individually, let us stay for a moment longer in this gentle place, this threshold between chemistry and life. In the world of non-living matter, molecules exist, but they do not self-replicate. They do not build cells. They do not breathe, grow, or repair themselves. But in the living world, a quiet miracle occurs. Simple molecules, when arranged just so, gain the ability to carry instructions, to fold and twist into functional shapes, to serve as messengers and builders and guides. In this way, the molecules of life are not just matter, they are also meaning. They carry information through the spirals of DNA and the loops of RNA. They store energy through the tiny, high-energy bonds of ATP. They shape the contours of every cell membrane, and they act as the catalysts behind every heartbeat, every breath. To understand molecules is to understand the language of life, spoken at its quietest volume. You don't need to memorize anything tonight. Just allow the words to drift softly through your awareness, like molecules floating in a gentle current. Let us now consider the scale of this world. Molecules are incredibly small. A single drop of water contains more than a septillion molecules, a number so large it escapes ordinary comprehension. And yet, each one plays a role. Each one obeys the same physical laws. Each one holds together through bonds that are at once strong and fragile. Despite their size, molecules are not uniform in shape or behavior. Some are simple, like oxygen, made of just two atoms. Others are intricate and folded, like the proteins that twist into complex forms to carry out specific tasks in cells. And some, like DNA, stretch into spiraling ladders that can store the code for an entire living being. All of this, all of life, begins with molecules. As we move through this soft biology journey together, you'll encounter many familiar words: glucose, protein, enzyme, DNA. Perhaps you've heard these terms before, in science class or in health articles, but here, they are not trivia. They are part of a gentle story, a story about how the smallest structures quietly make life possible. There is no urgency here, no test to take, no race to finish, just a quiet unfolding of knowledge, as calm as the night. And so, as you drift further into rest and your breathing deepens, let your thoughts gently settle on this idea that your body, at every moment, is quietly alive with molecules, not by accident, but by elegant design. Their motion is not random. Their form is not arbitrary. Each molecule in you was shaped by ancient forces, refined over billions of years, selected not by intention, but by quiet success. And in this silence, you are part of that continuity, part of the same molecular symphony that animates every leaf, every fish, every bird, every breath of life. In the next part of our journey, we will meet the quiet hero of this molecular world, the element called carbon. For now, let this thought guide you into stillness. That life, in all its wonder, is made possible by molecules. To the elegance of carbon. As you continue to rest, gently settled in stillness, allow your attention to float deeper into the heart of life's quiet chemistry. At the center of nearly every biological molecule lies a single, unassuming element: carbon. It is not the most abundant element in the universe, nor the heaviest, nor the most reactive, but it is the most versatile. And in that quiet versatility, it becomes the thread that weaves all living matter together. To understand why carbon is so essential to life, we must begin, as always, with its structure. A single atom of carbon contains 6 protons in its nucleus, 6 neutrons, and 6 electrons orbiting gently in invisible shells. Of these electrons, 4 are found in the outer shell, a place chemists call the valence shell. And it is here, in this outermost space, that carbon's gentle magic begins. Because carbon has 4 valence electrons, it can form 4 bonds with other atoms. This may seem like a small detail, but it is profound. With 4 bonding sites, carbon can link to 4 different atoms at once, or form double and triple bonds when needed. It can make long chains, closed rings, and branching networks. It can bond to other carbons, to hydrogen, to oxygen, to nitrogen, and to sulfur. This ability to bond in multiple directions to form stable yet flexible frameworks makes carbon the ideal backbone for the vast complexity of biological molecules. No other element offers quite the same elegant combination of strength, adaptability, and stability. Carbon can form small molecules, like methane, a single carbon with 4 hydrogen atoms, a simple tetrahedron quietly floating in space. Or it can form vast, intricate molecules like the spiral ladders of DNA, the delicate loops of enzymes, or the flexible membranes that cradle each living cell. Every sugar, every fat, every protein, every strand of genetic code begins with carbon. Even you, as you lie here in quiet rest, are composed in large part of this element. Carbon weaves through your tissues and organs, carried in your blood, nestled in your bones, and softly embedded in the very architecture of your cells. It is the reason we call life on Earth carbon-based. And yet, carbon's story is not confined to living organisms. In the natural world, carbon is found in rocks, in oceans, and in air. It drifts through the atmosphere as carbon dioxide. It rests in ancient shells and stones. It even forms precious solids like graphite and diamond, both composed entirely of carbon, but arranged in different ways. One is soft and dark, the other hard and crystalline. A quiet reminder that even within sameness, variation can bloom. In the world of biology, carbon rarely works alone. It pairs often with hydrogen, a small and abundant element, with oxygen, which participates in breathing and energy, with nitrogen, which builds amino acids and nucleic acids, with phosphorus, a keeper of energy, and with sulfur, a subtle shaper of protein form. These combinations, called functional groups, are like attachments that lend personality to each molecule. They determine how a molecule behaves, what it can bind with, whether it is water-loving or water-avoiding. A hydroxyl group, for instance, makes a molecule more hydrophilic, more willing to interact with water. An amine group adds nitrogen, often giving rise to amino acids. These small touches, these quiet additions, help shape the function of every molecule in your body. Carbon does not just build, it gives shape, function, and in some cases, even memory. The most famous of these memory-holding molecules is DNA deoxyribonucleic acid, which carries the instructions for building all the proteins in your body. Its elegant double helix, made of carbon-based rings, stores information as a sequence of nitrogenous bases, a soft code written in chemical form. That code tells each cell how to grow, how to divide, how to repair, how to be. In this way, carbon carries not only matter, but meaning. When life first emerged on Earth in warm pools or near hydrothermal vents, it was likely carbon compounds that came together first. Small molecules, like formaldehyde or hydrogen cyanide, may have bonded and recombined, forming sugars, amino acids, and lipids. These molecules, over time, assembled into self-replicating systems, a first whisper of life in the stillness of early Earth. In essence, life is carbon. And tonight, as you rest, your body continues to carry on this quiet choreography. Within each cell, carbon atoms are rearranged, broken down, built up, passed along. They form new molecules. They store and release energy. They shape your breath, your movement, even your thoughts. Beyond your body, carbon flows through nature as part of the carbon cycle. Through the air, it moves as carbon dioxide. Through plants, it is drawn in and turned into sugar. Through animals, it moves again, eaten, absorbed, breathed out. And when organisms die, carbon returns to the soil, where it may linger for years or re-emerge through decay or combustion. It is a closed loop. And so, the same carbon atoms that once flowed through ancient forests, through long extinct animals, through seashells and stones and stars, may now be flowing through you. This is the unity of life, a single element, dancing through forms. It does not speak, and yet it tells a story. Carbon silent, stable, shape-shifting, is life's essential framework. And as you drift further into rest, let this thought cradle your awareness, that within you and all around you, the quiet work of carbon continues, forming bonds, building structure, carrying memory, a soft foundation, an ancient participant, an invisible thread. In the next part of our journey, we'll meet water, the gentle solvent, the silent enabler of carbon's creativity. But for now, let the elegance of carbon settle softly in your thoughts and carry you deeper into calm. 3. Water. The silent medium of life. Let us now turn, with gentleness, to a molecule that flows quietly through every living being. Water. It fills your cells, cushions your organs, carries nutrients, and regulates your temperature. It moves through you, around you, and before you, ancient and unassuming. Life, in every known form, depends upon water not just as a backdrop, but as a participant. And not just a participant, but the stage, the solvent, and the silent conductor of biology's countless reactions. More than half of your body is composed of water. A newborn baby may be nearly 3/4 water. Even the hardest tissues, bone and cartilage, hold water within their matrix. It fills the spaces between cells. It slips across membranes. It flows through vessels and vacuoles, bringing with it dissolved minerals, oxygen, and warmth. But what makes water so uniquely suited to life? The answer begins, as always, with its structure. A water molecule is composed of 2 hydrogen atoms and 1 oxygen atom, H2O, arranged in a gentle angle, not a straight line, but a bent shape, like a soft pair of wings. And because of this structure, a quiet asymmetry arises. The oxygen atom holds the shared electrons a bit more tightly than the hydrogens. As a result, one end of the molecule carries a slight negative charge, while the other end is slightly positive. This is what scientists call a polar molecule, one with a split in its electric charge. And from this gentle polarity, many of water's remarkable properties emerge. Water molecules are drawn to each other. The positive hydrogen end of one molecule is softly attracted to the negative oxygen end of another. These attractions, called hydrogen bonds, are not rigid or permanent, but rather like a gentle magnetism, forming and breaking as water flows. This gives water an unusual cohesion. It clings to itself, forming droplets, beads, and films. It allows water to travel upward in plants, from root to leaf, against the pull of gravity. This quiet cohesion also explains why water has such a high surface tension, why it can support the weight of an insect, or ripple with the touch of a finger. Water also adheres, it sticks to other substances, to glass, to metal. This adhesive nature, combined with its cohesion, allows water to climb narrow tubes, to moisten soils, to move gently through tissues. Perhaps most astonishing is how water moderates temperature. Thanks to its hydrogen bonds, water resists sudden changes in heat. It takes a great deal of energy to warm it, and just as much to cool it. This is why oceans temper the climate of coastal lands, why your body remains stable even in fluctuating weather. Water's high specific heat means it can absorb warmth during the day and release it slowly at night. This buffering property creates comfort, not only in nature, but within you. Your body's temperature remains steady, thanks largely to water. And when water evaporates, when it slips from liquid to gas, it carries heat away with it. This is why sweat cools your skin, why mist feels refreshing. A quiet release, a hidden exchange. Water is also a universal solvent. Its polar nature allows it to dissolve more substances than any other liquid. Salts, sugars, amino acids, gases, all can become part of its flow. This dissolving ability is why blood can carry nutrients, why cells can exchange chemicals, why digestion, respiration, and circulation can occur. Imagine now the molecules dissolved in your bloodstream, calcium ions, glucose molecules, oxygen, each surrounded by gentle shells of water, buffered, transported, delivered. It is not forceful. It is not loud, just calm, diffusion, and soft movement. Even inside a cell, water creates the environment in which enzymes function, in which proteins fold, in which signals are sent. Cells themselves are enclosed in membranes, and these membranes, composed of lipids, rely on water's polarity to maintain their shape. The hydrophobic parts turn inward, away from water, while the hydrophilic parts face out, drawn to it. In this way, water not only flows through life, it shapes it. It creates boundaries and enables passage. It dissolves waste and supplies nutrients. It cradles the embryo, floats the leaf, fills the jellyfish. It is within and around all things that live. On the grand scale, water links ecosystems from rainfall to rivers to oceans. It carves valleys and fills lakes. It supports coral reefs, rainforests, and wetlands. It exists in three states: solid, liquid, and vapor, shifting with the seasons and the sun. In the water cycle, it rises from sea and soil, condenses into clouds, falls as rain, and returns again. A silent, endless motion, as soft as breath. And within your body, a similar cycle unfolds. You take in water through food and drink. It is absorbed through your digestive tract, drawn into your bloodstream, shared with your organs. It is used, recycled, and eventually released through sweat, through breath, through urine, and tears. This, too, is a quiet loop, a gentle exchange, an ongoing rhythm. Water is not simply a substance. It is a medium, a memory holder, a quiet collaborator in every moment of being alive. Without it, life could not exist as we know it. With it, molecules find their partners, reactions unfold, energy is released, waste is cleared, and life flows onward. As you continue to rest, you might imagine water moving through your body now, silently, smoothly, caring for each cell, cooling, cleansing, carrying. And as you breathe, remember that every inhale carries water vapor, that the air itself holds moisture, and that even in the driest places, water finds its way. We are, in a very real sense, beings of water and carbon, a gentle union of fluid and form. In the next part of our lecture, we'll meet the four major classes of biological molecules, the large, essential groups that water and carbon help bring to life. But for now, you may remain here, floating softly in the idea of water, the silent medium of life. 4. Four classes of biomolecules. As you rest deeper and your body surrenders to the quiet rhythm of sleep, we continue our soft exploration of the molecules that make life possible. So far, we've met carbon, the flexible backbone of organic chemistry, and water, the fluid medium through which life unfolds. Now, we step into the gentle world of life's major building blocks. Though all living organisms are profoundly complex, their structures and functions arise from just a few types of large molecules, known collectively as biological macromolecules, or simply biomolecules. Despite their variety, these biomolecules belong to just four major families: carbohydrates, lipids, proteins, and nucleic acids. Each class serves a unique and essential purpose, and together they compose the quiet architecture of cells, the machinery of metabolism, and the memory of heredity. Let us meet them, one at a time. We begin with carbohydrates. At their simplest, carbohydrates are sugars, sweet, crystalline, water-loving molecules made from carbon, hydrogen, and oxygen. These sugars can exist alone, as in the single ring of glucose, or joined together in small chains, like sucrose, or in great branching networks, like starch or cellulose. Carbohydrates serve as fuel. When you eat a piece of fruit or a grain of rice, your body gently breaks it down into glucose. That glucose then flows into your bloodstream, enters your cells, and is used to make ATP, the energy molecule that powers everything from your heartbeat to your thoughts. But carbohydrates are also more than food. In plants, long chains of sugar molecules form cellulose, the structural fiber that stiffens stems and strengthens leaves. It is one of the most abundant organic molecules on Earth. And yet, in your own body, it passes through undigested, a silent traveler that supports digestion by adding bulk. From energy to structure, carbohydrates are life's first quick answer to movement and growth. Next, we meet lipids. Unlike carbohydrates, lipids do not mix easily with water. Their molecules are nonpolar, smooth, and hydrophobic, preferring to cluster together, away from water's embrace. This trait gives lipids their essential roles. Fats and oils store energy in compact form, more than twice the energy per gram compared to carbohydrates. When your body runs low on sugar, it slowly turns to these lipid reserves, tucked away in soft tissues, waiting quietly. But lipids also serve as barriers. Every cell in your body is enclosed by a lipid bilayer, a double sheet of phospholipids, arranged so that their water-loving heads face outward and their water-fearing tails turn inward. This arrangement creates a cell membrane, flexible, self-healing, and selectively permeable. It's what allows cells to keep their inner environment stable, while still communicating gently with the world around them. Some lipids are hormones, chemical messengers that float through your blood, whispering instructions to distant tissues. Steroids, such as cholesterol and estrogen, are lipids too, shaping everything from your development to your emotions. So while they may seem subtle, lipids shape how life stores energy, builds boundaries, and sends messages. The third group is perhaps the most varied: proteins. These are the workhorses of the cell, the builders, movers, and facilitators. Proteins are made from smaller units called amino acids. There are 20 standard amino acids, and the way they link and fold gives rise to an endless variety of shapes. Some proteins act as enzymes, speeding up chemical reactions that would otherwise take far too long. Others serve as transporters, like hemoglobin, which carries oxygen through your blood. Still others form muscle fibers, hair, antibodies, hormones, or receptors embedded in cell membranes. Every protein begins with a chain, but that chain coils, bends, and folds into a shape, and it is that shape that defines its role. If the shape is lost through heat, or pH, or mutation, the protein may stop working altogether. This quiet dependence on form is part of biology's deep elegance. Function follows structure, and structure follows a code, which brings us to the fourth group, nucleic acids. These are the molecules that carry information. DNA deoxyribonucleic acid stores instructions for building all the proteins in your body. It resides in the nucleus of each cell, wrapped in protective spirals, quietly copied when new cells are made. RNA ribonucleic acid helps carry those instructions from the nucleus to the rest of the cell, where proteins are built. These molecules are composed of nucleotides, small units that link into long chains. The order of these nucleotides acts like a language, one that spells out the sequences of amino acids needed to build proteins. A soft genetic script written in a four-letter alphabet, passed from parent to offspring, unrolling quietly over generations. Together, these four groups, carbohydrates, lipids, proteins, and nucleic acids, form the fabric of all life. They are not confined to humans, or animals, or even plants. Every bacterium, every fungus, every coral reef, every leaf and seed is built from these same molecules. They differ only in arrangement, in proportion, in design, but the ingredients remain the same. And so, as you sleep, and your body gently repairs and restores itself, these molecules continue their work. Sugars deliver energy, lipids maintain boundaries, proteins build and rebuild, and nucleic acids hold the memory of who you are. All this complexity and beauty begins with just a few molecular families. Quiet, steady, essential. In the next part of our lecture, we'll drift more deeply into the first of these groups, carbohydrates, and explore how sugars, in all their forms, support and shape the living world. But for now, you may let these four names rest in your thoughts. Carbohydrates. Proteins. Nucleic acids, the quiet chorus of life's molecular song. 5 carbohydrates, energy and structure. As you lie still, letting your thoughts dissolve into the softness of the moment, we now turn our attention to the quiet sweetness of life's simplest fuel, carbohydrates. The very name comes from a soft pairing of elements, carbon, hydrogen, and oxygen, joined in balanced proportion. Often, the formula follows a gentle pattern, a carbon atom, a water molecule, repeated in harmony. It is from this pattern that carbohydrates earn their name, carbon plus water. But these are not mere numbers on a page. They are the source of your warmth, your motion, your breath. Every time your muscles move, every time your mind drifts through a memory or a dream, somewhere within you, a carbohydrate has quietly given its energy. Carbohydrates begin in simple forms, monosaccharides, small, single sugar units. The most well-known of these is glucose, a molecule with 6 carbon atoms, folded into a gentle ring, each atom holding hands with hydrogen and oxygen. Glucose is life's universal fuel. Whether you are a hummingbird or a human, a tree or a bacterium, glucose powers your cells. It travels easily through your bloodstream, reaching cells across your body. Once inside, it enters a series of careful reactions, a pathway known as cellular respiration, where it is gently dismantled, one bond at a time, releasing energy in the form of ATP. ATP, in turn, carries that energy to wherever it is needed, to contract a muscle, to send a nerve signal, to grow, divide, or heal. Other simple sugars include fructose, found in fruits, and galactose, found in milk. Though similar to glucose, each has its own subtle shape, its own role in taste, digestion, and metabolism. When 2 monosaccharides join together, they form a disaccharide. Sucrose table sugar is one such pair, a gentle union of glucose and fructose. Lactose, the sugar in milk, is a bond between glucose and galactose. These disaccharides are slightly larger, but still sweet, still soluble, still a ready source of energy. Beyond these, sugars can link into longer chains, oligosaccharides, or into vast, branching polymers called polysaccharides. These polysaccharides are no longer sweet, and they no longer dissolve so easily. Instead, they become structure and storage. In animals, Glucose chains form glycogen, a soft, branching polymer stored in the liver and muscles. When your blood sugar drops, your body can break down glycogen into glucose again, providing a steady reserve, a quiet wellspring of energy between meals. In plants, the storage form is called starch. It too is made of glucose units, linked in winding spirals and loops. When you eat potatoes, grains, or legumes, the starch within is slowly broken down into simple sugars, fueling your cells, hour by hour. But some carbohydrates serve not as food, but as scaffolding. Cellulose, another glucose polymer, is the most abundant organic compound on Earth. It forms the cell walls of plants, giving shape to stems, leaves, and roots. Though we cannot digest cellulose, the bonds are arranged in a way our enzymes cannot unlock. It still plays a quiet role in our bodies. As fiber, it moves through the digestive tract, supporting the motion of digestion, sweeping gently through the intestines, aiding in regularity and health. In this way, even undigested sugars contribute, not with energy, but with structure and flow. Carbohydrates also adorn the surfaces of cells, short sugar chains attached to proteins and lipids on the outer membrane, forming glycoproteins and glycolipids. These small decorations serve as identifiers, helping cells recognize each other, communicate, or respond to signals. It is a soft language, a molecular handshake, spoken in sugars. In the vast world of biology, carbohydrates are everywhere. They form the cell walls of bacteria and fungi. They make up chitin, the tough exterior of insects and crustaceans. They appear in mucus, in connective tissue, in breast milk. They shape taste, texture, and nourishment. They are among the first molecules we encounter in life, from the sugars in amniotic fluid, to the lactose in mother's milk, to the starch in warm, cooked grains. And they remain with us through every breath and heartbeat as a quiet, sustaining presence. Some carbohydrates are complex and slow to digest, offering long-lasting energy. Others are quick and fleeting, like a whisper of sweetness, but all share the same basic form: carbon, hydrogen, oxygen, arranged into gentle rings or chains. No matter the form, their story is the same: to provide energy, to build structure, to facilitate connection. And so, as you lie here in stillness, perhaps you can imagine these sugars flowing gently through your body, through your blood, entering your cells, releasing warmth and motion. Each molecule, no larger than a speck of dust, carries the potential to lift an arm, to smile, to dream. In the next part of our lecture, we'll move into the world of lipids, the smooth, quiet molecules that form boundaries, store energy, and whisper messages across cells. But for now, you may rest in the sweetness of carbohydrates, soft molecules of energy and form, flowing through every living thing. 6 lipids, silken molecules of storage and structure. Now, with your body deeply at ease and your mind floating gently in stillness, we continue our soft journey through life's quiet chemistry. In the world of molecules, some move freely in water, dissolving, mixing, and flowing with ease. Others prefer to keep their distance. Lipids are among these molecules that do not blend into water, but instead drift separately, forming soft layers, droplets, and boundaries. They are the silken shadows of biology. Quiet, hydrophobic, essential. At first glance, lipids may seem simple. They are made, like carbohydrates, from carbon, hydrogen, and oxygen. But the arrangement is different: more carbon, longer chains, fewer oxygen atoms. These differences give lipids their unique nature: smooth, nonpolar, and water-fearing. Because of this, lipids behave differently than sugars. Instead, they cluster away from it, like oil separating from vinegar, or like tiny droplets forming pearls on the surface of still water. This quality, their gentle resistance to water, gives lipids many of their roles in living systems. Let us begin with the most familiar form. A fat molecule is formed from a single glycerol backbone, linked to 3 fatty acid chains. This structure is called a triglyceride. The fatty acids themselves are long chains of carbon and hydrogen, unbroken, smooth, and winding. Some fatty acids are saturated, meaning they contain only single bonds between the carbon atoms. These tend to be solid at room temperature, like butter or lard. Others are unsaturated, with one or more double bonds that create gentle bends in the chain. These remain liquid, like olive oil or sunflower oil. When you eat fats, your body digests them slowly, releasing energy gradually, storing the excess in specialized cells called adipocytes. These cells do not just store fat, they cushion organs, insulate the body, and hold energy in reserve for times of need. The energy stored in fat is dense, more than twice that of carbohydrates, making it an efficient, long-term source of fuel. But lipids are not just fuel. They are also structure. Every living cell is surrounded by a membrane, a thin, flexible envelope that defines its boundaries. This membrane is built from phospholipids, special lipids, with 2 tails and a single head. The tails are hydrophobic. They avoid water. The heads are hydrophilic. They are drawn to water. When phospholipids are placed in water, they arrange themselves into a bilayer, a double sheet with tails facing inward, heads facing outward. This arrangement forms the cell membrane, a soft yet sturdy layer that separates the inner world of the cell from the outside. Within this membrane, proteins float, receptors and channels that control what enters and exits, signals that guide communication, all gently cradled within the lipid sea. Lipids create not just the skin of cells, but also the wrapping around organelles, tiny compartments inside cells. They form vesicles, small bubbles used to carry materials. They shape the flow of life inside and between cells. Among lipids, there is another group, more compact, more specialized. These are the steroids, a family of lipids built not from chains, but from rings. The most familiar steroid is cholesterol. Often misunderstood, cholesterol is essential. It is woven into cell membranes, where it adds stiffness and flexibility. It also serves as a building block for steroid hormones. These hormones, such as estrogen, testosterone, and cortisol, are chemical messengers. They travel through the blood, passing quietly into cells, where they bind to receptors and gently influence growth, mood, development, and balance. Other lipids serve protective roles. Waxes, for instance, coat leaves and feathers, preventing water loss. In your ears, a waxy lipid mixture forms a quiet barrier against dust and bacteria. Still others participate in signaling. Some lipids act like whispers between cells, brief messages released during inflammation, healing, or response to the environment. Though they move slowly and resist water, lipids are not passive. They are deeply involved in shaping life. They decide what can pass through a membrane. They hold energy in reserve. They protect and insulate. They carry signals, and they support the form of every cell. Like a gentle sculptor, lipids mold the landscape of the living world. They are in every membrane, every nerve sheath, every droplet of oil, every hormone that flows with purpose. And though they are often unseen, they are essential to the softness and stability of life. As you lie here, perhaps you can imagine them now, lining each cell, floating in membranes, coiling in reserve, drifting silently in blood. They do not shout. They do not force. They support. they shape, they soften. In the next part of our lecture, we'll meet the most dynamic of biomolecules, proteins, the folded structures that carry out life's many tasks. But for now, you may rest in the quiet knowledge that lipids soft, smooth, and patient are with you always, cradling your cells, storing your energy, and forming the silent architecture of your form. 7. Proteins. Folding. Function. Into form. Folding. As the stillness deepens and your breath settles into its gentle rhythm, we arrive at a new chapter in our journey through the molecules of life. So far, we have visited the flowing sweetness of carbohydrates and the silken calm of lipids. Now, we open the quiet door to a world of astonishing variety and complexity, the world of proteins. Proteins are not just structures or fuels. They are the workers of the cell, the silent performers of nearly every task life requires. They build, they repair, they move, they signal, they protect, they catalyze, and they do so with grace. Shaped by their form, guided by invisible instructions, and folded into delicate precision. To understand proteins, we begin with their smallest unit, the amino acid. There are 20 standard amino acids used in life. Each is a small, simple molecule, sharing a common structure, a central carbon atom, bonded to 4 groups, a hydrogen atom, an amino group, a carboxyl group, and a unique side chain called the R group. This R group varies from one amino acid to another, giving each its character. Some are small, some are bulky, some are water-loving, others water-fearing, some carry a charge, some are neutral. Like letters in a language, they are distinct, but when combined, they create meaning. A protein begins as a string of amino acids joined one by one in a specific order, determined by the code written in DNA. This sequence is called the primary structure, a simple chain, like beads on a string. But this is only the beginning. Once the chain is formed, it begins to fold, softly and precisely. First, it coils and bends into local patterns, spirals called alpha helices, and sheets, known as beta-pleated sheets. This is the secondary structure, shaped by hydrogen bonds between neighboring parts of the chain. Then, the entire molecule folds further, twisting into a unique 3-dimensional shape, its tertiary structure. Here, the R groups interact, some attracting, some repelling, forming bridges, pockets, and grooves. In some cases, multiple protein chains join together, forming a quaternary structure, a soft assembly of subunits that work as one. It is in this final folded shape that a protein finds its function. For a protein is not just a string of parts, it is a shape, a surface, a tool. Its form determines what it can do, what it can bind to, what reaction it can perform. Change the shape, and the function may be lost. This is why proteins are so sensitive to heat, to pH, to chemical change. A gentle rise in temperature or a small shift in acidity can cause the structure to unravel. This is called denaturation when a protein loses its shape, and with it, its ability to function. But when folded correctly, proteins are astonishing in their diversity. Some act as enzymes, speeding up the chemical reactions of life. Without enzymes, most reactions in your body would take minutes, hours, or longer. With enzymes, they occur in milliseconds. They are catalysts, silent accelerators, specific, and efficient. Each enzyme binds to its substrate, the molecule it acts upon, like a key fitting into a lock. It performs its task, releases the product, and begins again. Other proteins act as transporters, carrying molecules where they are needed. Hemoglobin, for instance, carries oxygen through your bloodstream. It binds oxygen in the lungs, releases it in the tissues, and returns to pick up more. Some proteins are structural, forming the shape of cells and tissues. Collagen strengthens skin and bone. Keratin makes up hair and nails. Actin and myosin slide past each other to make muscles contract. Others act as hormones like insulin, which helps regulate blood sugar, or as antibodies defending the body from invaders, or as receptors, sitting in cell membranes, receiving signals from hormones, neurotransmitters, or light. Even within the same cell, thousands of different proteins may be present, each folded just so, each with its own purpose, and though they are made from just 20 amino acids, the combinations are nearly endless. A short protein might contain 50 amino acids, a long one, thousands. With each position offering 20 choices, the number of possible proteins is beyond imagining. Yet your cells and the cells of every living organism know which ones to make. They read the instructions written in DNA, transcribe them into RNA, and translate them into proteins, one after another, precisely, silently, and once formed, proteins set to work, folding, moving, reacting, coordinating the breath of life. You are composed of proteins. They flow in your blood. They hold your cells together. They send signals through your nerves. They blink your eyes and heal your wounds. They are not static. They are always in motion, being built, being broken down, being recycled, a soft, continuous renewal. As you rest here now, you might imagine these folded structures within you, so small, yet so essential, spirals and sheets, globules and strands, each one crafted from a simple chain of amino acids. They do not speak, yet they carry out the tasks of life without pause, without fatigue. In the next part of our gentle lecture, we'll explore the remarkable world of enzymes, a special class of proteins that speed up life's chemistry, guiding every reaction with quiet precision. But for now, let your mind drift with the proteins, the silent architects and engineers, the folded forms that make life happen. 8 enzymes, catalysts of life's reactions. Let us now soften our focus and slip into the calm world of enzymes, quiet, precise, and endlessly devoted to their task. As you rest, the cells within your body are not still. They are alive with subtle motion, a continuous rhythm of building and breaking, combining and separating. And all of this activity, all the reactions that keep life gently unfolding, would be far too slow without the silent presence of enzymes. Enzymes are a special class of proteins. But unlike structural or signaling proteins, enzymes have one primary role. They serve as catalysts. A catalyst is something that speeds up a chemical reaction without being used up in the process. And enzymes do this with quiet elegance, guiding molecules together, holding them in just the right position, and lowering the energy required for the transformation to occur. They are the facilitators of life's chemistry, neither forcing nor pushing, only easing, aligning, allowing. Each enzyme is highly specific. It is shaped to fit one particular substrate, the molecule it acts upon, and only that substrate. This fit is often compared to a lock and key. The enzyme is the lock. The substrate is the key. And only the correct key can turn within that lock. Some enzymes act quickly, binding a substrate, rearranging it, and releasing the product in milliseconds. Others take more time, forming a series of subtle steps, folding, aligning, reacting, until the task is complete. Once finished, the enzyme is unchanged. It is ready to begin again, to quietly repeat its role, as many as thousands of times per second. This remarkable ability to act quickly, precisely, and repeatedly makes enzymes essential for life. Without them, the chemical reactions inside your body would be too slow to sustain you. A single step that now takes a fraction of a second might instead take years. And yet, with enzymes present, these steps flow seamlessly, one after another, like a soft, silent dance. Let us look, for a moment, at the process more closely. When an enzyme meets its substrate, the two come together at a special place on the enzyme's surface, known as the active site. This site is shaped just right, allowing the substrate to settle in. And once it does, subtle changes may occur. The enzyme may bend slightly, like a hand curling gently around an object. This is called the induced fit, a small adjustment that makes the interaction even more precise. Inside the active site, the bonds of the substrate are placed under gentle strain, aligned just so, encouraging the reaction to begin. Sometimes the enzyme helps break a molecule apart. Other times, it helps join two molecules together. It may add a phosphate group, remove a hydrogen, shuffle atoms into a new arrangement, and when the reaction is finished, the enzyme releases the product and returns to its original form, ready to guide the next reaction. There are many types of enzymes. Digestive enzymes help break down the food you eat, turning proteins into amino acids, carbohydrates into sugars, fats into fatty acids. In your saliva, an enzyme called amylase begins breaking starch into smaller sugars. In your stomach, pepsin breaks proteins apart. In your small intestine, other enzymes finish the job, each with its own task, its own timing. Inside cells, enzymes help build new molecules, copy DNA, repair damage, produce energy, detoxify waste. They regulate metabolism, ensuring that reactions occur only when needed and never too fast or too slow. Some enzymes even work in groups, pathways, where the product of one reaction becomes the substrate of the next. Like a quiet assembly line, each enzyme plays its part, passing the molecule along, transforming it step by step into something new. Enzymes also respond to signals. Some are turned on or off depending on the conditions, the pH, the temperature, the presence of other molecules. This regulation ensures balance, so that cells do not waste energy, so that reactions do not spiral out of control. Enzymes are not rigid machines. They are flexible, responsive, living tools. Their activity is shaped not just by structure, but by context, by the environment they work in, and by the needs of the cell. And just like all proteins, enzymes are made from amino acids, folded into specific shapes. If that shape is lost through heat, chemical change, or mutation, the enzyme may no longer function. This is why fevers can be dangerous, why pH must be carefully maintained, why a single change in a gene can alter an entire pathway. But when enzymes work as intended, the effect is profound. Thousands of chemical reactions, each one precise, each one necessary, occurring within you every second, silently, faithfully, without pause. You do not feel them. You do not direct them. But they continue soft and constant like waves in a tide. And so, as you rest, your body is not idle. Within you, enzymes are guiding molecules together, speeding reactions, building and breaking in perfect balance. You are, in this quiet way, always renewing. In the next part of our gentle exploration, we'll turn to nucleic acids, the molecules that carry life's memory and hold the instructions for every protein ever made. But for now, let yourself be still and imagine these folded catalysts, small, precise, endlessly patient, helping life unfold again and again. 9. Nucleic acids, molecular memory. As your breath deepens and your body softens further into calm, we arrive at the next chapter of life's molecular story, a story not of structure or energy or speed, but of memory. Long before there were books or language or even conscious thought, life found a way to remember itself, to pass knowledge forward not in words but in molecules. This memory lives in the form of nucleic acids, the silent record keepers of biology. There are two main types of nucleic acids: DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. These molecules are composed of long, delicate chains of smaller units, known as nucleotides. Each nucleotide contains three parts: a sugar, a phosphate group, and a nitrogenous base, a ring-shaped structure that holds the key to life's instructions. In DNA, there are four types of bases: adenine, thymine, cytosine, and guanine, often shortened to A, T, C, and G. These bases pair in a specific and predictable way. Adenine always joins with thymine, and cytosine with guanine. Like gentle puzzle pieces, they fit together and form the steps of DNA's iconic spiral staircase, the double helix. Picture that shape now. Two strands gently coiled, like ribbons twisting in space. Along their length, the base pairs reach across to embrace one another, A with T, C with G, forming a ladder of information. The sides of the ladder, made from sugars and phosphates, hold it all together in a soft, flexible frame. The order of the bases carries information, not unlike the letters in a sentence. This molecular script spells out the instructions for building all of the proteins in your body. These instructions are stored in sections of DNA called genes. Some genes are short, others are long, but each one is a quiet message, passed down from one cell to the next, from one generation to another. DNA is tightly coiled and stored in the nucleus of nearly every cell. When your body needs to use the information, it doesn't touch the original, it makes a gentle copy. This is where RNA comes in. RNA is similar to DNA, but more flexible, more temporary, and single-stranded rather than double. It carries the message of a gene from the DNA in the nucleus to the machinery of the cell, the ribosomes where proteins are assembled. This process, from DNA to RNA to protein, is known as the central dogma of molecular biology. It is how form becomes function, how code becomes chemistry. The ribosome reads the RNA 3 bases at a time. Each triplet is called a codon, and each codon corresponds to a specific amino acid. These amino acids are joined together, one by one, to form a protein. In this way, nucleic acids give rise to the entire machinery of life, from enzymes to muscle fibers to the hemoglobin in your blood. But RNA is more than just a messenger. Some RNA molecules help form the ribosome itself. Others carry amino acids to where they're needed. Some RNA acts like a switch, turning genes on or off depending on the needs of the cell. Even certain viruses, like influenza and coronavirus, carry their genetic information not in DNA, but in RNA, showing how this molecule, though simple in shape, can carry the spark of life. The information stored in nucleic acids is remarkably enduring. Some stretches of DNA have been copied over and over for millions, even billions of years, passed through fish, through mammals, through primates, into humans. With every generation, the message continues, softly edited, gently refined, but always connected to what came before. As you lie here now, in quiet rest, that message continues within you, coiled inside each nucleus, encoded in each gene. The soft script of DNA whispers the instructions for every cell. It doesn't need to be read all at once. It waits, patient, silent. When the time is right, a gene is copied, the RNA is formed, and a new protein takes shape, and with it, a new action, a new moment of life. You are, in this way, a living memory, a vessel of molecular instructions, shaped by the long line of life that came before you, from the first simple cell to your own gentle breath. In the next part of our soft exploration, we'll meet ATP, a molecule shaped not to remember, but to deliver. A silent courier, carrying energy from one reaction to another. But for now, let yourself drift with this thought that inside you, always, are the molecules of memory, quiet, coiled, and enduring. 10 ATP. The energy messenger, as you remain in stillness, drifting deeper into quiet, we arrive at a molecule so small, so simple, and yet so essential that every heartbeat, every breath, and every flicker of thought depends upon its presence. This molecule is known as ATP, adenosine triphosphate. While DNA carries the instructions and proteins perform the actions, ATP serves as the courier between them. It does not build, it does not record, it does not fold, but it does one thing perfectly, patiently, endlessly. It delivers energy. Every cell in your body requires energy to function, to move substances across membranes, to contract muscles, to send signals, to build molecules, to divide, grow, and repair. But this energy cannot be left scattered or uncontrolled. It must be packaged, measured, and delivered precisely. That is the quiet role of ATP. The structure of ATP is composed of 3 parts. First, a nitrogenous base called adenine, the same adenine found in DNA and RNA. This is attached to a 5-carbon sugar called ribose. Together, they form adenosine. To this core, 3 phosphate groups are attached in a row. It is in the bond between these phosphate groups, especially the final one, where the energy is stored. When a cell needs energy, it breaks the bond, holding the last phosphate group. This process, called hydrolysis, turns ATP into ADP, adenosine diphosphate, and releases 1 phosphate group, along with a small but usable burst of energy. This energy is used instantly, powering everything from muscle movement to the chemical steps of metabolism. Once ATP is used, it is not discarded. The cell quietly restores it. A phosphate is reattached to ADP, reforming ATP again. This cycle creation, use, regeneration, happens endlessly. A single cell may produce and consume millions of ATP molecules each second, and your body, in its entirety, recycles its own weight in ATP every day. The place where most ATP is made is within your mitochondria, the small organelles known as the powerhouses of the cell. As food is broken down during cellular respiration, electrons are released. These electrons move through a series of proteins in the mitochondrial membrane, creating a flow, an electric-like gradient. This movement powers an enzyme that builds ATP. It is a quiet current, an invisible wind, that fuels all life inside you. ATP is not only made in the mitochondria. Some is produced in the cytoplasm, through a simpler process known as glycolysis. In plants, ATP is also formed during photosynthesis, capturing energy from the sun and storing it in molecular form. Across all forms of life, whether in a human heart cell or the leaf of a tree, ATP quietly carries the spark of life. Beyond its role as an energy currency, ATP can act as a messenger. It helps open ion channels, trigger muscle contractions, and regulate cell signaling. Yet always, Its core task remains the same: to carry energy from one molecule to another, releasing it only when needed. Imagine it now, if you wish. ATP molecules floating gently inside you, delivering soft pulses of energy to every cell. They do not shout. They do not pause. They simply arrive, offer, and return to begin again. In this way, ATP supports all living motion, softly, silently. Without it, life would falter. Muscles would go still. Signals would stop. Healing would cease. But with it, the smallest tasks, a blink, a breath, a heartbeat, continue effortlessly. And so, as you rest now, your cells continue their work. Energy flows. ATP is spent, renewed, and spent again. You are at rest. And yet, life within you continues steady and gentle, carried by this tireless messenger. In the next part of our journey, we will begin exploring the living cell, the tiny community of molecules and structures where all of this unfolds. 11. The living cell, a molecular community. As you continue to rest, gently wrapped in the stillness of sleep, we arrive at the quiet chamber where all of life's molecules come together, the cell. It is here, within this tiny, self-contained space, that the sugars, lipids, proteins, nucleic acids, and energy carriers, like ATP, all meet, each with its role, each in balance. The cell is the smallest unit of life. It is not a hollow shell, nor a simple sack of fluid. It is an intricate, living system enclosed in a thin boundary, filled with organized parts, and animated by a flow of gentle chemical events. Whether it is a single-celled bacterium or one of the trillions of cells in the human body, each cell is complete in itself, self-regulating, self-repairing, and quietly alive. At its most basic level, every cell is surrounded by a soft, flexible membrane, a lipid bilayer that defines its shape and separates its inner environment from the outside world. This membrane is not solid, but fluid. Its surface is dotted with proteins, some acting as gates, others as sensors, others as quiet markers of identity. This outer boundary allows the cell to control what comes in and what goes out: oxygen, water, nutrients, signals. Inside, the cell is filled with a substance called cytoplasm, a watery gel that holds all of the cell's inner parts, or organelles. Though mostly water, the cytoplasm contains salts, proteins, sugars, and countless molecules floating, folding, and interacting in silence. It is in this space that life's chemistry unfolds, not chaotically, but with structure and rhythm. In the center of most cells lies the nucleus, a round, membrane-bound compartment that stores the cell's DNA. This is the control room, the library, the memory center. Inside the nucleus, genes are copied into RNA. That RNA then travels outward, carrying the instructions for building proteins, instructions that will be read and followed in precise detail. Surrounding the nucleus and extending throughout the cell are networks of folded membranes known as the endoplasmic reticulum. There are two types, smooth and rough. The rough ER, speckled with ribosomes, is where proteins are assembled. The smooth ER handles the creation of lipids and detoxifies substances, softly maintaining balance within the cell. Floating nearby are the ribosomes themselves, tiny structures made of protein and RNA. These are the molecular factories that build proteins, reading the code carried by messenger RNA and linking amino acids one by one, folding them as they go. Despite their size, ribosomes are vital. Without them, no protein could be made, and life would quietly pause. Next, we find the Golgi apparatus, a stack of flattened membranes that receives proteins from the ER, modifies them, and sends them to where they're needed. Like a post office, the Golgi carefully sorts and packages these molecules. placing them into tiny vesicles that travel to the membrane or deeper into the cell. Perhaps the most remarkable of the organelles are the mitochondria, the powerhouses of the cell. These bean-shaped structures have their own membranes, their own DNA, and their own gentle rhythm. Within their inner folds, nutrients are broken down, electrons are passed along, and ATP is formed, the quiet, glowing currency of energy that powers every task the cell performs. Other organelles include lysosomes, which contain enzymes that break down worn-out parts, and peroxisomes, which quietly detoxify substances. There are also cytoskeletal fibers, long, thread-like proteins that stretch across the cell, providing shape, movement, and support. They are not rigid, but dynamic, growing, shrinking, shifting as needed. In plant cells, there are chloroplasts, green organelles, where sunlight is captured and converted into sugar, and large central vacuoles filled with water and nutrients, creating pressure that supports the plant's structure. Each part of the cell works not in isolation, but in connection with the others. Messages pass, materials move, enzymes guide reactions, and all of it flows under the soft governance of DNA and the gentle supply of ATP. Even in the simplest cells, like bacteria, though they lack a nucleus and many organelles, bacteria still possess membranes, cytoplasm, ribosomes, and DNA, a quiet framework of life, ancient and enduring. And so, the cell, though small, is a complete world. Within it, molecules do not float aimlessly. They act with purpose. They are guided by shape, by charge, by pattern. Every cell is a self-sustaining system, capable of reacting to its environment, dividing to grow, and passing along its instructions. As you rest now, trillions of your cells are at work. Some are forming proteins, others are repairing membranes, some are sending signals down nerves, others are digesting nutrients, or dividing to replace the old. All of them are connected, working softly, quietly alive. In the next part of our journey, we will explore the cell membrane in more detail, that gentle boundary where communication, protection, and exchange all begin. But for now, let yourself rest in the comfort that within you, each cell is its own peaceful world, a community of molecules working together in silence and in balance. 12. The cell membrane, guardian and gatekeeper. Now that we have gently stepped inside the cell, it's time to pause and look again at the boundary, the quiet interface between life and the world beyond. This is the cell membrane, thin, fluid, and nearly invisible. It is the guardian of the cell, a flexible gatekeeper that surrounds and protects the internal world of every living cell. Though only a few nanometers thick, the cell membrane is richly complex. It is not a rigid wall, nor a simple wrapping. Instead, it is a dynamic, living surface made primarily of phospholipids, arranged into what we call the phospholipid bilayer. Each phospholipid has two parts, a head that loves water, and two tails that shy away from it. When placed in water, these molecules arrange themselves naturally. The heads face outward, toward the watery environments inside and outside the cell. The tails tuck inward, away from the water, forming a double layer, soft, sealed, and self-healing. This structure forms the basic shape of the membrane, a gentle envelope that flexes, curves, and shifts, but rarely breaks. Floating within and upon this lipid sea are proteins, each with its own role. Some proteins serve as channels, allowing water or ions to pass through. Others act as carriers, changing shape to move larger molecules across. Some are receptors, receiving signals from outside and passing messages inward. Others form anchors, connecting the cell to its surroundings or holding the membrane in place. These proteins do not drift randomly. Many are positioned with purpose, responding to the needs of the cell, its environment, and its function. In a nerve cell, for example, proteins help transmit electrical impulses. In an immune cell, they recognize foreign invaders. In all cells, they help maintain balance, allowing nutrients in, waste out, and signals to pass gently through. Scattered along the outer surface of the membrane are carbohydrate chains, attached to proteins and lipids. These sugar structures form a kind of molecular signature, identifying the cell to others. They help with recognition, communication, and protection, much like a name tag or a soft handshake. Each cell displays its own unique pattern, shaped by its role in the body. The cell membrane is also involved in movement. When large particles or fluids must enter the cell, the membrane gently folds inward, forming a pocket that encloses the material. This process is called endocytosis. Similarly, when the cell needs to expel something, it creates small vesicles that merge with the membrane and release their contents, a process known as exocytosis. Despite its flexibility, the membrane also plays a role in maintaining homeostasis, the stable internal conditions needed for life. By carefully controlling what enters and exits, the membrane helps regulate temperature, pH, nutrient levels, and waste, allowing the cell to function without interruption. In multicellular organisms, the membrane also allows for cooperation. Cells can connect to one another through special junctions, sharing resources or sending signals. Some membranes form tight seals. Others allow small molecules to pass between neighboring cells. In tissues and organs, this coordination is vital, a quiet harmony that keeps the body whole. The membrane is also sensitive to the outside world. Changes in temperature, pressure, light, or chemical signals can be detected through its surface. These changes may trigger cascades of molecular responses within the cell, helping it adapt, respond, or defend itself. And though it is constantly active, the membrane remains soft, flexible, alive. Its molecules move laterally, like a quiet current. Some proteins are fixed in place, others drift. The lipids themselves can shuffle, rotate, and even flip from one side to the other, though such flips are rare and carefully controlled. In this way, the cell membrane is both stable and fluid. It preserves the identity of the cell, and yet allows it to grow, divide, change, and communicate. It is the interface between inner life and the outer world. Not a wall, but a window. Not a cage, but a quiet harbor. As you rest now, picture the cells within you, each one wrapped in its own living skin, soft, protective, and responsive. Through these membranes, nutrients are drawn in, messages are heard, and balance is quietly maintained. Without a single sound, your cells know when to open, when to close, and when to remain still. In the next part of our sleep-bound journey, we'll explore how molecules move through this membrane, how water flows, how nutrients travel, and how life maintains its rhythm through gentle transport. But for now, rest in the image of the cell membrane, delicate, vital, and always in motion, a guardian, a gate, and the quiet edge where life begins. 13 molecular transport, flow, and exchange across the membrane. Across the membrane, as your body remains still and your mind sinks deeper into quiet, we turn our attention now to the soft movement of molecules, the gentle traffic that flows across the cell membrane. Though the membrane forms a barrier, it is not closed, it is not silent. It breathes, it listens, it allows life to pass through in ways that are carefully controlled, yet beautifully simple. This flow of materials is known as molecular transport, and it takes several forms. Some are passive, requiring no energy, driven only by natural movement. Others are active, requiring the cell to spend energy, often in the form of ATP, to move substances in or out. And each method plays its part in sustaining the quiet rhythm of life. Let us begin with passive transport, the simplest and most peaceful form of movement. Imagine a drop of dye spreading through water. Slowly, gently, the color moves from where it is concentrated to where it is sparse. This natural spreading, driven by the random motion of particles, is called diffusion. In cells, many small molecules, like oxygen or carbon dioxide, move in this way. No force is applied. No energy is spent. They drift freely across the membrane, from areas of high concentration to areas of low concentration. Some substances, like water, move by a special kind of diffusion called osmosis. Water molecules pass through the membrane or through special channels called aquaporins, flowing toward regions where water is less abundant. This movement helps maintain the right balance of fluids, keeping cells from swelling or shrinking. Osmosis is quiet but constant, a molecular tide. Other molecules are too large or too charged to pass through the membrane freely. For them, the cell provides facilitated diffusion, a form of passive transport where proteins help guide the molecules across. These proteins act like doors that open only for specific shapes. Glucose, for example, enters cells this way, moving down its concentration gradient, helped along by a carrier protein. All of these methods, diffusion, osmosis, facilitated diffusion, occur without the use of energy. The cell simply allows molecules to follow their natural paths. It is the biology of balance, a movement toward calm. But there are times when the cell must move molecules against their natural direction from where there is little to where there is already much. This is called active transport, and it requires energy. One of the best-known examples is the sodium-potassium pump. This protein, found in most animal cells, moves sodium ions out and potassium ions in against their gradients. With each cycle, it uses one molecule of ATP, ensuring that the cell maintains the proper balance of ions. Though the movement is tiny, the effect is profound. It helps generate electrical signals, maintain volume, and support countless cellular activities. Some transport is even more dramatic. Large particles, such as fluids, nutrients, or bits of debris, are taken in through a process called endocytosis. Here, the cell membrane folds inward, wrapping around the particle and forming a vesicle that pulls it inside. The cell does not grab, it simply embraces, forming a soft pocket. In the reverse process, exocytosis, vesicles inside the cell merge with the membrane, releasing their contents to the outside. This is how hormones are secreted, how neurotransmitters are released, and how waste is quietly removed. Together, these methods, passive and active, allow the cell to exchange with its environment, to receive what it needs, to release what is no longer useful, and to remain, always, in a gentle state of dynamic balance. Molecular transport also plays a role in communication. Ions, flowing through channels, generate tiny electrical signals. In nerves and muscles, these signals move quickly, allowing for thought, motion, and feeling. But beneath that speed lies a softness, a steady exchange of atoms, through membranes sculpted by evolution, guided by shape and charge. As you lie still now, every cell in your body continues this transport. Oxygen enters your lungs, passes into your blood, and diffuses into your tissues. Nutrients absorbed in your gut are ferried across membranes. Water flows, ions move, and energy pulses, molecule by molecule. These movements are silent, invisible, yet they never stop. They are the tides of life, flowing through you, as gently as breath. In the next part of our lecture, we will turn to the language of charge and concentration, the world of pH, acids, bases, and buffers, and how the cell maintains the quiet chemical balance so essential for life. But for now, imagine your cells like small harbors, where ships come and go, molecules drift and settle, and currents flow just as they must, everything arriving at the right moment, everything leaving in peace. 14 pH and buffers. The chemistry of balance. As you drift further into rest, we now step into the quiet chemistry of balance, the subtle world of acids, bases, and the soft measurement we call pH. It is in this realm that molecules interact through charge, not shape. Here, the presence of a single atom, a lone hydrogen, can shift an entire system. And yet, even these tiny changes are guided, softened, held in place by nature's own caretakers. The buffers. The pH scale is a simple number, but it speaks of a deep equilibrium. It tells us about the concentration of hydrogen ions, also known as protons, dissolved in a solution. These ions are small but powerful. They are involved in nearly every biological reaction from protein folding to energy transfer. The more hydrogen ions present, the more acidic the solution. The fewer, the more basic. This scale ranges from 0 to 14, with 7 as the middle point neutral. Water, in its pure form, rests at this midpoint. Solutions with a pH below 7 are acidic, such as lemon juice or stomach acid. Solutions above 7 are basic, like baking soda or soap, but inside the body, the range is far narrower. Most cells function best at a pH just around 7.4, slightly basic, but very close to neutral. Even small deviations, a shift of just 0.2 or 0.3, can disturb proteins, disrupt enzymes, or interrupt communication. For this reason, the body works quietly, constantly, to maintain this fragile chemical balance. This is where buffers come in nature's gentle stabilizers. A buffer is a combination of molecules that can absorb or release hydrogen ions, depending on the situation. When the environment becomes too acidic with too many hydrogen ions, the buffer binds some of them, softening the shift. When it becomes too basic with too few hydrogen ions, the buffer releases some back into solution, restoring harmony. One of the most important buffering systems in the human body is the bicarbonate buffer, found in the blood. When carbon dioxide dissolves in water, it forms carbonic acid, which can release hydrogen ions. The body carefully controls this system through breathing and kidney function, keeping blood pH within a safe and steady range. Buffers exist inside cells as well, maintaining the pH of the cytoplasm, the nucleus, and the fluid surrounding each organelle. Even the stomach, which produces strong acid to digest food, protects itself with layers of mucus and localized buffering systems. Each environment has its own pH and its own way of staying quietly in balance. The importance of pH extends beyond biology. In soil, pH affects which nutrients are available to plants. In oceans, shifts in pH, often from rising carbon dioxide, can change the behavior of coral, plankton, and shell-forming creatures. Even rainwater, as it falls through the sky, absorbs gases and adjusts its pH. But in the body, the regulation of pH is constant, intimate, and essential. Enzymes only work at certain pH levels. Membranes are affected by charge. Ion channels open and close depending on the acidity of their surroundings. ATP production, protein stability, and nerve transmission all rely on the right concentration of hydrogen ions. And yet, through all of this, your body does not ask for attention. It does not raise alarms unless pushed too far. It simply adjusts. It breathes out carbon dioxide. It filters ions through the kidneys. It deploys buffers. And the pH, gently and persistently, remains where it should. As you lie here now, Your own cells continue this balance. Blood flows, carrying bicarbonate. The lungs release carbon dioxide with each exhale. The kidneys fine-tune the mixture of ions. All of this happens without noise, without tension. It is a chemistry of whispers, of adjustments made not in leaps, but in nudges. A drop of acid, a shift of base, and the buffer responds. In the next part of our journey, we'll explore the quiet choreography of metabolism, how cells break down and build up molecules to grow, heal, and thrive. But for now, rest in the knowledge that within you, even the tiniest ions are held in harmony, balanced, buffered, at peace. 15. Metabolism, the cell's gentle engine. As you lie still, perhaps drifting further into the softness of sleep, we now enter the flowing heart of cellular life, the world of metabolism. It is not a single process, but a quiet network of reactions, unfolding gently, continuously, within each of your cells. Metabolism is the name we give to all the chemical transformations that occur inside living organisms. It is the cell's way of managing matter and energy, breaking down molecules to extract fuel, and building new ones to grow and repair. Though the word may sound mechanical, metabolism is anything but forceful. It is delicate, rhythmic, a choreography of molecules, guided by enzymes, fueled by ATP, and shaped by need. At its core, metabolism is divided into two complementary branches, catabolism and anabolism. Catabolism is the breaking down, the disassembly of larger molecules into smaller ones. It is how the body digests food, how cells extract energy, how old or damaged molecules are gently taken apart and recycled. When glucose, fats, or proteins are broken down, they release energy. This energy is not wasted or lost. It is captured in molecules like ATP, stored in silence, ready to be used. Within catabolism lies the soft fire of cellular respiration, a process where glucose is gradually dismantled. It begins in the cytoplasm with a pathway called glycolysis, where one molecule of glucose is split into two smaller pieces. A few ATP molecules are made here, a quiet beginning. Then, if oxygen is present, the products of glycolysis enter the mitochondria, where the citric acid cycle begins, a circular pathway that gently strips away electrons from carbon compounds, releasing carbon dioxide as a byproduct. These electrons are passed through a series of proteins in the electron transport chain, creating a flow that powers ATP synthesis. It is here, in the folds of the mitochondria, that most of the cell's energy is born, not in bursts, but in waves. Each glucose molecule yields dozens of ATPs, all formed in careful sequence. And when oxygen is low, cells may turn to fermentation, a slower, less efficient path, but one that allows survival. Muscles, for instance, produce lactic acid during intense activity. Yeast produces ethanol. Each pathway reflects the flexibility of metabolism, its willingness to adapt, to continue, even when conditions change. Meanwhile, on the other side of this balance is anabolism, the building up. In anabolic pathways, cells use energy to construct complex molecules. Amino acids are linked into proteins. Fatty acids are joined to form lipids. Sugars are combined to make polysaccharides. Nucleotides become strands of DNA. These processes require ATP gentle deposits of energy and careful guidance by enzymes. Anabolism is growth. It is healing. It is renewal. Even now, as you lie and rest, your cells are quietly rebuilding. Skin cells divide. Proteins are repaired. Bones strengthen. Blood regenerates. This soft maintenance never ends. What is most remarkable is how metabolism remains balanced. The cell senses what it needs. It reads internal cues, levels of ATP, concentrations of certain molecules, signals from hormones, and responds. When energy is abundant, catabolic processes slow, and anabolism begins. When resources are scarce, the cell conserves, prioritizing survival. This balance is managed by enzymes, feedback loops, and gentle molecular switches. Some enzymes are activated by the very molecules they help produce. Others are turned off when their products become too abundant. In this way, the cell never does too much or too little. It works just enough, and then rests. Across the body, this metabolic rhythm plays out in every organ. The liver breaks down toxins and stores glucose. The muscles burn energy to move. The brain, though small, consumes a quarter of your fuel, using it to think, to remember, to dream. Even in sleep, your metabolism continues slower, steadier, but unceasing. Some organisms, like plants, reverse the cycle during the day. Through photosynthesis, they use sunlight to build sugars from carbon dioxide, and water, a gentle anabolic process powered by light. Animals, in turn, consume those sugars. Life's energy flows in circles. As you lie here now, your own metabolism flows quietly. Fuel is broken down, energy is made, molecules are built, all of it in balance. Nothing rushed, nothing wasted. In the next part of our journey, we will explore the proteins that guide this metabolic dance. The enzymes, revisited now with deeper insight, as masters of molecular motion. But for now, feel free to drift deeper, knowing that even as you sleep, your cells are tending their own slow fire building and breaking in harmony. A quiet engine, ever turning, ever gentle. 16 enzymes revisited. Masters of molecular motion. As we float deeper into the quiet symphony of life, it is time to return to a familiar presence, the enzymes. We met them earlier, in their role as catalysts, guiding reactions with silent precision. But now, with a fuller view of the cell's internal world, we can appreciate their role in even greater depth, not merely as helpers, but as the quiet conductors of molecular motion. Enzymes are proteins, long chains of amino acids folded into unique, three-dimensional shapes. It is their shape that gives them purpose, their form that determines their function. Each enzyme has a specific task, and only that task to speed up a single chemical reaction by guiding it along the smoothest path. In a world where chemical change could take years or longer, enzymes make it happen in fractions of a second. But what makes enzymes so remarkable is their efficiency. They do not force reactions. They do not push or pull. Instead, they bring molecules together in just the right way, at just the right time, to lower the energy needed for transformation. This threshold is called activation energy, and enzymes lower it like a soft slope carved into a steep hill, allowing molecules to glide where they once struggled to climb. Within the enzyme is a small region called the active site, a pocket, a groove, a gentle fold where the substrate binds. The substrate is the molecule the enzyme will act upon. Once the substrate settles into place, the enzyme may subtly shift, adjusting its shape in what is called an induced fit. This gentle embrace aligns the atoms, weakens certain bonds, and invites the reaction to proceed. Some enzymes help break molecules apart. Others join them together. Some rearrange atoms, or transfer chemical groups, or form temporary complexes with other molecules. Whatever their role, enzymes are not changed by the reaction. When the task is done, they release the product, return to their original form, and begin again. Within a single cell, thousands of enzymes work in harmony. Each is assigned to a specific reaction in a larger metabolic pathway, a chain of chemical steps that leads from raw materials to useful products. The product of one enzyme becomes the substrate for the next, forming a soft, flowing sequence of transformations. These pathways are not random. They are organized, regulated, and adaptable. Some are linear, others are cyclical, like the citric acid cycle we visited earlier. In each case, enzymes are the guides, ensuring that the right reactions happen at the right time. Enzymes are also regulated by the cell. Their activity can be turned up or down, activated or inhibited, depending on conditions. A process called feedback inhibition allows the end product of a pathway to gently shut down the very enzyme that began it, ensuring that the cell does not overproduce. This is not abrupt, but balanced, a quiet loop that preserves resources. Some enzymes require cofactors to function, small helper molecules, like vitamins or metal ions, that assist in binding or catalysis. Others are part of larger complexes, working together like a coordinated machine. And while enzymes are incredibly efficient, they are also fragile. Their shape depends on temperature, pH, and surrounding conditions. If the environment shifts too far, the enzyme may denature, losing its structure, and with it, its function. This is why balance is so important in the cell, why pH is regulated, why heat must be controlled. In nature, enzymes shape everything from digestion to DNA replication. In your saliva, enzymes begin breaking down carbohydrates. In your stomach, others dissolve proteins. In your cells, they copy genetic information, build membranes, and clear waste, all without you ever noticing. Even now, as you lie in stillness, your enzymes are at work, repairing DNA, clearing toxins, assembling proteins. They do not tire, they do not ask, they simply fold into place, bind, release, and begin again. And so, enzymes are more than tools. They are the silent conductors of life's chemistry, subtle, precise, and deeply patient. In the next part of our lecture, we'll explore how all of these molecules, enzymes, substrates, energy carriers, and more come together in networks of biochemical pathways, forming the intricate flow of reactions that sustain all living systems. But for now, rest in the knowledge that within each of your cells, enzymes are guiding molecules, with grace turning chaos into calm, speed into rhythm, reaction into harmony. 17 biochemical pathways, the flow of cellular life. Now that we have met the enzymes, the quiet facilitators of change, we step into the soft, branching networks they create. These are the biochemical pathways, not straight lines, but intricate threads of interaction that weave the full tapestry of cellular life. Each path is purposeful, each reaction feeds into the next, and together, they form the hidden rhythm of the living cell. A biochemical pathway is a series of linked reactions, each step guided by a specific enzyme. The product of one reaction becomes the substrate for the next, passed forward like a soft baton in a molecular relay. These pathways do not drift aimlessly. They are organized, regulated, timed with extraordinary care. Some of these pathways break things down, releasing energy and reducing complexity. These are the catabolic pathways, like glycolysis, which gently splits glucose into smaller molecules, or the citric acid cycle, which processes those fragments within the mitochondria. Together, these reactions generate energy, stored in the bonds of ATP, carried to where it is needed. Other pathways build, creating structure, storing information, or preserving energy. These are the anabolic pathways. They assemble amino acids into proteins, fatty acids into lipids, sugars into polysaccharides. They construct DNA and RNA, repair membranes, and form the molecules of communication. Many of these pathways are cyclical, like a soft loop of reactions, constantly turning. The urea cycle removes nitrogen waste. The Calvin cycle in plants converts carbon dioxide into sugars. The citric acid cycle, which we've already visited, spins endlessly in the presence of oxygen, releasing energy at each step. Some pathways are branched, with intermediate molecules flowing in different directions depending on the cell's needs. One product may be used to build a protein, Another may be burned for energy. These branches give the cell flexibility, allowing it to adapt, to prioritize, to respond. And between these larger systems are the regulatory mechanisms, subtle molecular signals that turn reactions on or off. An abundance of product may slow a pathway. A shortage of a starting material may speed it up. Enzymes may be activated by molecules binding at distant sites, or by changes in the cell's environment. In this way, biochemical pathways are self-correcting, guided not by thought, but by feedback. They keep the cell from overreacting or wasting energy. They ensure that resources are used wisely, and that every task is completed at the right time, in the right place. These pathways are not confined to one cell. They connect across tissues, organs, and systems. The liver breaks down toxins and sends glucose to the muscles. The muscles use it for motion, releasing carbon dioxide. The lungs remove that carbon dioxide, returning oxygen. The digestive system absorbs nutrients, and the bloodstream carries them to where they are needed. All of it is coordinated. All of it flows. Even the hormones that signal hunger, growth, or rest are themselves products of biochemical pathways. They are synthesized, modified, packaged, and released in response to subtle changes, forming another layer of gentle regulation. And though each pathway may seem small, the total number is immense. A single human cell may have thousands of enzymes, guiding tens of thousands of reactions every second. And yet, it does not feel chaotic. The cell does not rush. It does not panic. As you rest now, your own cells are following their pathways. A molecule of glucose is entering glycolysis. A strand of RNA is being transcribed. A protein is folding in the endoplasmic reticulum. None of it needs direction. The instructions are encoded in the molecules themselves, in their shapes, their charges, their affinities. It is chemistry shaped by memory, action guided by structure, and life expressed through motion. In the next part of our journey, we will gently shift outward to the organelles once more, this time focusing on their interactions and communication, like neighbors in a quiet community. But for now, let yourself drift with this image. A soft network of reactions flowing within you, unbroken and ongoing, as gentle and constant as your breath. 18 organelles in harmony, a community within. As you remain nestled in stillness, your breath quiet and your body at ease, we return once more to the hidden world inside every cell. But now, with clearer eyes, we no longer see only isolated parts, nucleus, mitochondria, ribosomes, but rather a community. Each organelle, though distinct, works in close relationship with the others. Together, they form a silent network of cooperation, a harmony of function, unfolding every moment within the life of the cell. Imagine now the nucleus, round and central, gently enclosed in a double membrane. This is the cell's library where DNA is stored, protected, and copied. From here, instructions flow outward in the form of RNA. These soft strands drift into the cytoplasm, carrying the genetic message that tells the cell what proteins to make. Waiting in the cytoplasm, or nestled on the surface of the rough endoplasmic reticulum, are the ribosomes, the small, patient builders. They read the RNA like a quiet script, translating it into amino acid chains. These chains begin folding into the specific shapes that define each protein's role, a process as intricate as it is silent. The endoplasmic reticulum, with its folded membranes, serves as a highway and a factory. The rough ER hosts protein assembly. The smooth ER, by contrast, produces lipids, detoxifies harmful substances, and stores calcium, essential for many types of signaling. It is smooth in appearance, but deeply active. Newly formed proteins are then delivered to the Golgi apparatus, a stack of flattened pouches where they are gently modified, sorted, and prepared. The Golgi adds sugars or other chemical groups, packages proteins into vesicles, and sends them to their destination, perhaps to the membrane, perhaps to another organelle, or perhaps to be released from the cell entirely. Meanwhile, the mitochondria work continuously, absorbing nutrients and transforming them into ATP, the energy currency used by all other organelles. These mitochondria are not only powerhouses, they also coordinate signals for cell growth, division, and even self-repair. Their inner membranes fold into quiet chambers, maximizing surface area for the soft orchestration of energy. Elsewhere in the cell, lysosomes float small, enzyme-filled spheres that break down waste, recycle parts, and help defend against invaders. They are not destructive, but cleansing. Carefully contained by their membranes, they ensure that digestion occurs only where it should. Peroxisomes, too, assist with detoxification, converting harmful substances into safer forms, often with the help of oxygen. They also play a role in breaking down fats, forming a part of the broader metabolic network. At the cell's edges, the plasma membrane interacts with the outside world, receiving messages, taking in nutrients, and maintaining balance. Signals received at the surface are passed inward, often triggering cascades of events that ripple through the entire cell. Threaded throughout this space is the cytoskeleton, a flexible framework made of protein filaments. These filaments give the cell its shape, assist with transport, and help position each organelle. Vesicles move along these filaments like cargo on rails, directed with care to their proper location. Each organelle relies on the others. Proteins made in the ribosomes require energy from mitochondria. Mitochondria depend on lipids built in the ER. The Golgi needs both to modify and distribute molecules properly. Lysosomes and peroxisomes depend on enzymes produced elsewhere. Even the membrane's surface proteins are crafted within the cell, shaped and delivered through this soft and continuous collaboration. There is no central controller. No organelle gives orders, and yet everything happens in concert. Feedback loops, molecular signals, and shape-based interactions allow each part to know when to act, when to pause, when to adjust. This is the gentle logic of the cell, not top-down but emergent, not rigid but responsive. As you rest now, this community within you continues its work. Your cells do not sleep, even as you do. They renew membranes, digest waste, fold proteins, and generate energy. Each task completed without friction. Each interaction soft, precise, and quiet. In the next part of our journey, we will look beyond the single cell and into the origins of these molecules and how life, in its earliest days, may have begun. But for now, imagine your cells as small cities, each one alive with peaceful motion, each organelle like a neighbor, every task shared, every part in balance, a community within you, working in quiet harmony. 19 Origins of Biomolecules, Life's Molecular Dawn. As the body lies quiet and the thoughts grow soft, we now turn our gaze back far back to a time before cells, before organisms, before even the first stirrings of life. In this stillness, we drift to the ancient Earth, more than 3.5 billion years ago, where the first biomolecules may have formed. It was a world without people, without forests, without sound. The atmosphere was different, filled with volcanic gases, ammonia, methane, water vapor, and carbon dioxide. The oceans were warm, rich in minerals, stirred by tides, rain, and the flicker of lightning. From this early Earth, under skies streaked with storms and sunlight, the molecules of life began to emerge. Scientists call this the prebiotic Earth. the time before biology, and though many of the details remain unknown, there are theories, gentle, compelling ideas that describe how the first building blocks of life might have formed. In one such vision, simple molecules such as water, methane, hydrogen, and ammonia were exposed to energy from lightning or ultraviolet sunlight. This energy caused bonds to break and reform, creating more complex molecules. In laboratory simulations, this process has been shown to produce amino acids, the building blocks of proteins, and even nucleotides, the units of RNA and DNA. These early molecules did not appear all at once. They likely formed over long stretches of time, accumulating in shallow pools or near hydrothermal vents on the ocean floor. There, in those warm, mineral-rich spaces, reactions may have occurred more frequently. The Earth itself may have served as a quiet crucible, gently mixing, warming, and guiding molecular evolution. Over time, lipids, simple fat-like molecules, may have spontaneously formed bubbles or spheres in water, creating boundaries not unlike cell membranes. These primitive bubbles could trap other molecules inside, forming a soft division between the internal space and the outside world. A first step toward compartmentalization. At some point, molecules emerged that could copy themselves. Perhaps it was RNA, a molecule that, unlike DNA, can both store information and catalyze reactions. This dual ability may have allowed certain RNA strands to survive, replicate, and evolve. This is sometimes called the RNA world hypothesis, the idea that life began not with cells, but with strands of RNA floating in ancient seas, slowly refining their ability to persist. From this quiet beginning, metabolism may have followed simple reactions that extracted energy from the environment, and over time, as molecules became more organized, they may have joined within membranes, forming the earliest protocells. These were not yet alive by modern definitions, but they had structure. They could grow. Perhaps they could divide. And in their simplicity, they held the promise of life. No one knows exactly when chemistry became biology, when motion became metabolism, when molecules became memory. But somewhere along this ancient journey, the first living cell arose. It was likely small, simple, and surrounded by a membrane. It carried genetic information. It could reproduce. And from it, all life has descended. That earliest cell gave rise to bacteria, to archaea, to the ancestors of all multicellular life. Over time, cells learned to cooperate, to specialize, to form tissues, organs, and entire organisms. But the molecules remained the same: amino acids, sugars, nucleotides, lipids, the original players, still central to the story of life. Even now, every cell in your body carries echoes of that ancient chemistry. The ATP your mitochondria produce uses a molecular mechanism that first emerged billions of years ago. The genes that guide your development are made of the same nucleotide bases as those found in early RNA strands. The membrane that surrounds your cells shares structure with the lipids that once floated in the oceans of early Earth. Life, in all its diversity, remains deeply connected to its molecular dawn. The past lives quietly within us, in our chemistry, in our structure, in our every breath. And so, as you rest in stillness now, you are not separate from the story of life. You are part of it, a continuation of an ancient unfolding, a soft echo of a beginning that still whispers in every living thing. In the final part of our journey, we'll reflect on the unity of this molecular world, how all life, no matter how complex or simple, is built from the same gentle chemistry. But for now, let your thoughts rest beside those early tides, warm, quiet, and full of possibility. 20 Unity in chemistry, all life, one language. As we reach the gentle close of our journey, let us pause now, not to summarize, but to reflect. We have wandered through the quiet chemistry that builds every living thing from atoms to molecules to cells. We have watched as proteins folded, as DNA carried its silent code, as enzymes guided reactions with patient precision. And now, in the stillness of this moment, we look not outward, but inward, to the quiet truth that all life shares a common language, written not in words, but in molecules. From the simplest bacterium to the tallest tree, from coral in deep oceans to neurons in the human brain, every living thing is made from the same fundamental parts, the same nucleotides, arranged differently, the same amino acids folded into unique shapes, the same sugars joined into long or short chains, the same lipids drifting in membranes. These molecules do not belong to humans, or to animals, or to plants alone. They are shared, universal. A strand of DNA in a flower contains the same 4 bases as that in a whale. A protein in a mushroom is made from the same 20 amino acids as one in the human heart. The ATP that fuels your muscles is identical to the ATP used by a blade of grass to grow. The enzymes that copy genetic material in your cells bear deep similarity to those found in the first microbial life. This unity is not poetic, it is molecular. It is factual. It is deep. It suggests that all living things are connected not by chance, but by shared origin. We do not simply resemble one another in form or behavior. We are built from the same toolkit, shaped by time, by adaptation, by need. This is why cells from different organisms can sometimes accept genes from one another. It is why scientists can study fruit flies to understand human biology. It is why life in all its forms follows the same essential rules. Chemistry is the foundation, biology is the expression, and evolution is the story that links them together. And yet, within this shared language, there is endless variation. Just as music arises from the same notes, but forms infinite melodies, life composes itself in countless ways from the same molecular alphabet. It is not uniform, but diverse, not rigid, but adaptive. The building blocks remain the same, but the forms they take are wondrously wide. As you rest now, your own cells are speaking this ancient language. They are building proteins, repairing DNA, folding membranes, generating energy. Every second, trillions of molecules are moving through you, guided by forces that emerged long before you were born, and which will continue long after. And so, in this stillness, you are not apart from nature. You are a part of it, not as an observer, but as a participant. Every breath you take brings in atoms once held by stars. Every molecule in your cells has cycled through oceans, stones, and skies. You are not just made of molecules. You are part of a molecular lineage, unbroken, ancient, and alive. This is the quiet truth at the heart of biology. Not competition, not struggle, but shared chemistry, shared patterns, shared origins. And perhaps, as the night deepens and the world becomes more hushed, you may carry this calm with you, knowing that life is not loud, but soft, not scattered, but connected. That beneath the complexity of living things lies a single, elegant code, written in molecules, spoken without sound, understood by every cell. Let your thoughts settle now in this awareness of unity, of continuity, of peace, and sleep as life continues quietly within you,